This presentation is about a prenatal technique called amniocentesis. At the outset, I would like to give a warning that prenatal gender screening, that means to find out male-female child, has been banned, that is considered illegal in India. This restriction is put out of Prenatal Diagnostic Techniques Act 1994. Now, as we told, amniocentesis is a prenatal procedure to find out the possibility of the development of certain genetic diseases or disorders in the fetus developing in the body of pregnant mother. And this is done by taking a small quantity of amniotic fluid, along with which there may come some amount of fetal cells which would have been detached from the developing fetus. So the biochemical and chromosomal analysis of such fetal cells and amniotic fluid may give you some idea about the possibility of the development of such disorders in the fetus or the child to come. Now here this illustration may give you the position of the fetus inside the amnion of the pregnant mother. Now here you see the body skin part of the abdomen, and within the body wall there is the uterus, and inside the uterus there is the amniotic sac, and there is the amniotic cavity filled with the fluid called amniotic fluid, and which may have some of the cells would have been detached from the developing embryonic stage of the fetus when it was growing. And actually this fetus may be floating in the amniotic fluid, which would be a shock absorber and which would keep the fetus safe from the injuries caused due to the movement of the mother. So now here, after consulting the medical doctor or the genetic counselor, the need for amniocentesis, there the amniotic fluid is drawn out using a needle which would be very specific for this process. And then the amniotic fluid drawn out of this amniotic cavity. Here the care should be taken not to injure the fetus while the needle is inserted and the fluid is drawn out that way. About 20 ml said to be taken for the process of amniocentesis, because the total quantity of the amniotic fluid may be about 600 to 800 ml during the pregnancy of the mother. And then this amniotic fluid is centrifuged, and you will be having some fetal cells getting along with the amniotic fluid separate, and then the amniotic fluid. And by cell multiplication process you can multiply the fetal cells, and it may be subjected to the chromosomal analysis. And the amniotic fluid also may be subjected to some biochemical process. Now when due to the modern advancement in biotechnology, bioinformatics and microbiological procedures, you can find out even at the gene level any defect present by DNA screening methods. Now simply to say some examples, the present possibility for cystic fibrosis in the developing child, which would be the defect with the exocrine glands, and the muscular dystrophy, weakening of the muscles, even at the developing fetus or in the future. And the gene may cause the normal hemoglobin or sickle cell hemoglobin that way. That can be known. Even that way, the modern methods, possibility for the analysis of a variety of genes and by karyotyping to know the condition of the chromosome in a whole. You say one or two examples. If there the number 21 chromosome is a trisomy in condition, then the possibility for the Down syndrome, and then a Cat-Due-Chat syndrome if there occurs to be deletion at chromosome number 5. So these can be easily observed by karyotyping, and even by subjecting the amniotic fluid for biochemical analysis, like the content of AFP, which may be an indicator of the disease called spina bifida, nothing but the exposure of spinal cord from the neural tube of the developing fetus, and even the lung maturity of the fetus at the age of some 32 or 38 weeks may also be known through the analysis of this amniotic fluid. Even the presence of any infection may be known out of this. So that way a lot of possibilities for the deformities in the developing fetus may be identified, and